point out the demarcation here. Okay, so about this point, we no longer have an action potential moving from a voltage-gated sodium channel to another voltage-gated sodium channel, the way that we saw along the length of the axon. Okay, because we no longer have voltage-gated sodium channels here. And so this would not depolarize the membrane, would it? No. We need a voltage-gated sodium channel in order to fire an action potential. So the cessation of the voltage-gated sodium channels here is what stops the action potential from moving here and depolarizing the membrane here in terms of continuing that action potential being fired. I hope that makes sense. So the electrical energy of this action potential becomes chemical by exocytosing these neurotransmitters here. And that is facilitated by no longer having those voltage-gated sodium channels. If we don't have voltage-gated sodium channels, we don't have an action potential, right? Very important to make that distinction. Okay, so the action potential gets to the axon terminal. We open up these voltage-gated channels. Again, these are voltage sensitive. So when the action potential arrives, they will open up, okay? But in their opening up, they are not firing an action potential. They're doing a totally different thing. They are facilitating this exocytosis process. Okay, so although they're being responsive to that action potential, they are not resulting in another action potential. They're resulting in a chemical being released here. So the neurotransmitter then diffuses, it binds to its receptor on the membrane of the adjacent cell, and then we bring about some type of cellular response. Now, in order to cease or to terminate this signal, we must clear the junction. We must clear the synaptic cleft. Okay, so response will be terminated by removal of the excess neurotransmitter. Otherwise, we will continually bind neurotransmitter and continually fire signals, which is not what we want, right? We must clear the cleft. And so we can clear the cleft in one of three ways, which I sort of mentioned earlier. One is that we can break these down into smaller bits using this enzyme, and then it can diffuse away. Two is that it can diffuse straight away, right? Just in its natural state, those neurotransmitters simply diffuse away. And then three is that it can be reuptaken, repackaged, and then recycled, okay? Now there's a slight delay between when the action potential gets to the axon terminal and then the cellular response in the adjacent cell, about 0.5 to 5 milliseconds. And it's really important for us to explain what that delay is caused by. So when the action potential gets here, it's about 5 milliseconds before we see any sort of membrane potential change on this adjacent cell. And that is not, as it would be tempting of us to think that that's because it takes time for this diffusion to happen. And that's not the case, okay? That is not the case. So the actual explanation of the delay here is it takes time for the calcium to rush in. It takes time for this vesicle to make it down to the membrane, to dock onto the membrane, to fuse its membrane with the membrane of the axon terminal, and then for the neurotransmitter to be released, okay? So just to really highlight the importance here is that this is not the reason. The length of time of diffusion is almost instantaneous. So as soon as this neurotransmitter is dumped at the cleft, we see a response. The part of this that takes some time, the part of this that we have the delay is in the calcium coming in, the docking of those vesicles with the membrane here, and then the dumping or the exocytosis of the vesicles. Okay, so really important to make that distinction. Okay, so that is it for the synapse, what happens at the synapse. The most important part is making sure that you can distinguish those types of synapses, focusing more on chemical synapse, all right? Make sure that you underline, highlight that. That's the crux of everything else we're gonna talk about. And then looking at the steps, make sure you can elucidate those steps of the synapse. It's gonna be very important as we go on. Practice drawing them out, step 1, step 2, step 3, and so on. Next, we'll move on to what happens on that second cell. We're gonna compare two types of responses on the second cell, fast responses and slow responses, okay? And this is another very important area, guys. If you're an underliner or a highlighter like I am, make sure that you're double, triple, quadruple highlighting this topic. Very important, signal transduction. This is how we do a lot of physiologic processes in the body. Okay, the body's very efficient, and so it uses these pathways, these cascade of messengers. You tell someone to do something, he tells a second messenger, and that's how we relay a lot of signals in the body. So we'll speak about two types of responses. We'll talk about a fast response and then a more complicated slow response, okay? Now the fast response is sort of nice and easy because we have a channel that is linked to a receptor. And this is really the element of this that makes it fast. The channel is the receptor. Okay, so the channel acts as a receptor, and we have a ligand of some sort. Now, our ligand is going to be our neurotransmitter, and a ligand is nothing but a molecule or an ion that binds to something and causes a conformational change. So our ligand is our neurotransmitter, whatever that is, that will bind to this channel, which is also a receptor, and that will open up the channel, and it will facilitate ionic movement. So some type of ion X in question, will be able to move when this neurotransmitter binds. And that's just one way of bringing about a cellular response. Okay, so to sort of step back here, we're talking about what happens on that second cell. We just mentioned in our drawing that some type of cellular response was brought about, but how that cellular response is brought about is what we're going to move on to talk about now. Okay, so the fast version of this is that we have an ion channel, which is also a receptor. Question? So a neurotransmitter is, it's charged. It's a molecule that binds, it's very specific. There are lots of different types. There's GABA, there's glutamate, there's adrenaline. There are different types of neurotransmitters that have different properties, really. We're going to be speaking about adrenaline, norepinephrine as examples, GABA, glutamate, which are very different from each other, and they do different things. Okay, but it's a molecule that binds here. It's acting as a ligand, and that it's going to bring about some sort of change to this channel or change to this receptor. Okay, good question. Any other questions?
So are there specific receptors for, like you said, adrenaline?
Exactly. And that's what we're gonna talk about that in the autonomic nervous system. So we have adrenergic receptors, we have metabotropic receptors. Depending on what the neurotransmitter is, it's gonna have a very unique, specific receptor to bind to. Okay, we can have, for example, epinephrine can be a hormone in the blood, and then in the nerves, it's a neurotransmitter. Same molecule, same properties, but it becomes a neurotransmitter in that it can bind here and bring about cellular response. Okay, whereas in the blood, it's a hormone. So lots of different properties to different neurotransmitters. And so we're talking about the fast version of this response here, and this is quick and easy. The channel is going to close as soon as this neurotransmitter dissociates off. And the difference between being bound and being dissociated happens to be with the affinity, how much this neurotransmitter likes the receptor. And that can change depending on ion concentrations and other factors. So when it's time for this channel to close, the neurotransmitter becomes has less of an affinity for the receptor, and then it dissociates off, the channel closes. Okay, so this is the fast version of the response. Okay, now moving on to the slow response. So this is where things get a lot more interesting. Okay, these are called metabotropic receptors. I don't think I mentioned these. These are called ionotropic. So the fast receptors were called ionotropic. And then the slow receptors are called metabotropic receptors. These are slower in their response because these require a few middlemen, as I like to call them. These require a few messengers, a few people standing in between the receptor and the actual response. All right? The receptor is not the channel. It's not as easy and straightforward as ionotropic receptors because we have quite a few players in the middle here. So we have a receptor, again, very unique to its neurotransmitter, and then it's coupled to another protein here. And this protein, which has several subunits, is going to change its shape when the ligand binds this receptor. So the ligand, which is a neurotransmitter, binds to this unique receptor, which is coupled to this G protein, and the G protein has a conformational change such that one of the subunits here, specifically the alpha subunit, dissociates off, and it's going to go downstream in the membrane where there's another enzyme. We're going to talk about what these enzymes are called, but don't get too focused on the names at this point. Later on, you will have to know the names. But another enzyme downstream in the membrane, this alpha subunit attaches to that enzyme, activates that enzyme, and the enzyme is now turned on. It's going to start this second messenger, this sequence of events, this cascade of passing this message. The first thing it's going to do is create some sort of second messenger, usually cyclic AMP. We talked about cyclic AMP as one of our nucleotide, it's made up of one of our nucleotides, right? And so cyclic AMP is usually the second messenger involved here. And that's going to go on to do something else. At this point, we're not going to talk about what that is. Later on, we will. And that something else is going to bring about a cellular response. Okay, so as we run out of time, I'm really going to just drive home the main point, the main difference between these fast and slow responses. For slow responses, we have the receptor being the channel. So there's no middlemen, there's no messengers, there's nothing in between. When the neurotransmitter binds to that receptor, the channel opens up, we have flow of ions, okay? Let's say we have a sodium channel opening up. We have sodium rushing into the membrane, and we depolarize the membrane, and that's how we bring about that second response. So much faster, much easier, one structure involved. For metabotropic, or you have a question? Sorry? You had a question? No. So for the metabotropic or the slow version of the cellular response, we have quite a few messengers involved. Most importantly is this G protein that's coupled to the receptor. So the receptor is embedded in the membrane, and it's attached or coupled or bounded covalently to this G protein. And so when the ligand binds to the receptor, again, a unique or specific interaction, that changes this protein such that one of its subunits comes off and goes down the membrane. One of its subunits will be one of the messengers, right? One of the middlemen in this process. It goes down the membrane, and imagine that happening. It has to move around in the cell. This is why this is so slow, right? It has to dissociate off, move around in the cell, go to this enzyme, which is downstream in the membrane, bind to it, turn it on. This enzyme now starts making some other second messenger, and then the second messenger has to go and do something else. Okay, so this is a slower process, but, and I'll end here on this note, it's a much more amplified process. So this is going to bring about a bigger, more generalized response than the ionotropic response, okay? And we'll talk much more about